In this video, we will discuss about the gene editing method that can be used for in vivo CAR-T immunotherapy. So what is this in vivo CAR-T immunotherapy? This means that we don't have to make CAR-T in the lab. We can directly insert the instructions that helps in the formation of CAR-T within the body. So how these instructions are transferred? The instructions are usually transferred uh, in the envelope, which is commonly known as the virus. That is here the example shown here for this adeno associated virus. So here the CAR encoding DNA will be used, and the genes that they are going to target that is specific to the T cells is TRAC homologous sequence, or TRAC gene, that is T cell receptor alpha constant gene. And this is the map of this is a main component of the virus. That is, it should have this envelope, that is VSVG, that helps in the interactions with the cells, so that this virus can get internalized. It should have this Cas9 guide RNA as essential to edit the DNA, and the CD3 binding antibodies and EDB, these are also necessary so that it can interact with the cell surface of the patient's cells. So here the example shown here is that adeno-associated virus with the instructions containing the editing of these T cells will be transferred into the mice. The mice will see in vivo productions of CAR-T, and this how this CAR-T formations are occurred is that it will first target the T cells, because the T cells contains this targeted gene sequence, targeted gene, that is TRAC. It is T cells receptors alpha constant gene. So it can edit only this gene. So when it find out that gene in the T cells, then what it will does is it will cut this TRAC using the Cas9 guide RNA. And once it cuts this TRAC, then thereafter this TRAC homologous CAR encoding DNA sequence, which is also encoded, also provided in this virus. So this encoding DNA sequence will then be inserted into the TRAC, which has been cut by this Cas9 guide RNA. So thereafter we will have this CAR encoding sequence that replaces the TRAC. So this will enables the T cells to form this CAR encoding sequence, and since these CAR are designed to get expressed on the cell surface, they will be expressed on the cell surface. Once a CAR are expressed on the T cell surface, so this CAR has the ability to detect the cancer cells, and once it interacts with the cancer cells, it can trigger the cell death. So this is one of the gene editing method that can be used, which is specific to the T cells because it's targeting only TRAC genes which are expressed in the T cells. So this will enables to prevent less side effect or less editing on the other cells. However, there is also always a possibility that it can edit the other cells, because this is not 100% specific, although the specificity is high compared to the other gene editing tools that are currently being designed. And other things that one need to be careful is that this adeno-associated virus that they have used, it can also be modified in a way. Nowadays the specific adeno-associated virus are being designed to target specific organs, so we can use that capsid to insert this CAR encoding DNA and this TRAC homologous sequence along with this Cas9 guide RNA. So this will further enables the more specific editing on the T cells. So these things are currently under studies. This idea is currently under studies. However, some of the animal results are being published for using this gene editing in vivo CAR-T immunotherapy method. If you need to understand more about this gene editing method for in vivo CAR-T generations, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.